Good day, I am John Tapira from group number 3, and today I will be discussing about the microscopy, principles, parts, use, and care. On the second slide, you can see this picture of a microscope that has a corresponding names for each individual parts. And then on the third slide, we have here the ocular lenses. So ocular lenses is the eyepiece that magnifies the image produced by the microscope objective lenses for viewing. So basically, we observe these slides, prepared slides, or the specimens on the ocular lenses. Then we have here the diopter adjustment. The diopter adjustment is let the users adjust the focus of the eyepieces to their eyesight for a clear view of the specimen without needing a corrective lenses. So basically, diopter adjustment is a adjustment tool to adjust the focus of the eyepieces so that we could clearly see the specimen or the slide. Now, we have here the arm. So, the arm is the one that offers a structural support for the microscope. So, the arm is the one that links the head, body, and then the base. So, when we are properly holding the microscope, we are holding it on the arm supported by the base. Then, we have the nose piece. So, the nose piece or the turret is beneath the microscope. So, it holds all the objective lenses that have a different level of magnification. So, this allows the users to seamlessly turn or change the objective lenses without changing it manually. So, moving on the fourth slide, we have the objective lenses. The objective lenses is composed of multiple lenses that has a different level of magnification. So, this is the one that produce larger images serving various purposes such as extreme magnification and spatial filtering. So, objective lenses is the one that is used for magnification purposes on the slide so that we could view easily the slide or the specimen under the microscope. Then we have the spatial filtering. So the spatial filtering, by that definition, is the one that brings clarity on the image or the image of the specimen. So we have the stage clip. The stage clip is the one that holds the specimen on the stage. The stage is the platform of the microscope where the specimen or the prepared slide is being placed at. So we have the diaphragm. The diaphragm is the one that regulates the light coming from the light source, then to the diaphragm, then to the condenser, then finally we can view it through the ocular lenses. So we have the condenser. The role of the condenser or the function of the condenser is to collect the light that is coming from the illuminator or the light source and focus it on the specimen itself. The way that the condenser focus the light coming from the illuminator is in a cone shape. So through that, in a cone shape, it can easily focus the light on the specimen. So moving on, we have the coarse adjustment. So the function of the coarse adjustment is to bring the stage up and down for the rough focusing to bring the specimen into view. Then the fine adjustment. The fine adjustment is positioned near the coarse adjustment knob on the microscope. So it enables for the precise focusing to achieve clear and sharp image of the specimen by allowing subtle details of the stage for a refining focus and revealing finer details. So the fine adjustment is being used so that it could able to bring clarity to the specimen when it is viewed. Now we have the illuminator. So the illuminator or the light source is the microscope light source. It provides illumination from the below stage, enabling clear visibility of the specimen details under magnification. So this illuminator or light source can be made out of mercury or tungsten. Then moving on, we have the base. So the base is the one that supports the weight of the microscope. So it ensures the stability. It houses different circuits, illuminators, light switches, and electrical wiring system. Then we have here the light switch. So the light switch is usually found on the base. So it controls the illuminator by turning it on and off. When activated, the light passes through the aperture, slide, and objective lenses, illuminating the specimen. Its intensity can be adjusted for optimal viewing. So basically, the light switch is the one that controls the voltage, the flow of the voltage on the electrical wiring of the microscope. So it turns on and off when the user is using it. Then on the parasitology, the study of parasites, we have monocytes. It is found on the reproductive organs of the insects. The shape is varied forms depending on the life stage. The color is typically transparent or slightly colored. We have here the Trichinella spiralis larva, a parasitic roundworm that causes trichinosis in humans and animals. The shape is coiled and elongated. It has a translucent with whitish appearance. Then moving on, we have the Chilomastic mesnelli. It infects the human intestines and can cause gastrointestinal diseases. The shape is pear-shaped with a single flagellum, and then the color varies, but is very often that is colorless. Then moving on, we have the Hymenolopis, the Minuta gravid. It is a tapeworm that infects rodents and occasionally human. It is flattened, ribbon-like structure. The color is transparent or slightly opaque. Now we have the Dipylidium carcinum mature segment. It is a tapeworm that is primarily found in dogs and cats. And the shape is segmented and resembling a chain of rice grains. And then the color is usually pale and segmented. Now, we have the Gargia lambiasis. It is known to cause diarrhea and gastrointestinal infections in humans and animals. It has a oval or round shape with characteristics of nuclei. It is typically colorless or pale. On the histology or the study of the tissues, we have 4 different specimens. The first one is the mammal pyloric stomach section. It is characterized by pinkish to purple section displaying mammalian pyloric stomach, likely with glandular structures. And then the next one is the mammal fungiostomach. It is characterized by pinkish to purples made out of epithelial cells. Long elongated hair-like structure is observed, held by a basement membrane. It has like a marble-like appearance on the longitudinal section. Then the third one is the human bone decalcified. It has a light beige to pinkish on the longitudinal section of the decalcified human bone, emphasizing organic components like collagen. Then lastly, we have the cardiac muscle with Purkinje. It is striated appearance of the cardiac muscle. As we know, there is epicardium, pericardium, and endocardium. It has a distinct fibers like the Purkinje. It is responsible for the electrical signals that is coming from the AV node and as well as from the SA node to keep the heart from beating. On the slide number 19, we have the botany. The botany is the study of the plants, including their structure, growth, and reproduction. The first one we have on the prepared slides for the botany is the penicillium. The penicillium is a genus of mold often associated with the production of the antibiotic penicillin. The fungal structure is often filamentous with a color of black to dark when observed on the microscope. The next one is the spirulina. It has a nutritional value and use as a dietary supplement. The color is blue to green, spiral shaped cyanobacteria, and appearing bluish under the microscope. Then the third one is the pollen grains germinating. Germination providing a close-up view of the development of the pollen tubes. Microscopic view of germinating pollen grains, potentially showing elongated structures with colors depending on the staining, but on the specimen that is observed under the microscope, it has a dark color when it is observed on the microscope. The fourth one is the helianthus stem. It is used to study the histological features of stem tissue in this plant species, which is the sunflower. The cross-section of sunflower stem exhibiting vascular tissues and then a greenish color. Then the last one is the onion skin plasmolysis. It undergone shrinkage due to water loss. Plasmolyzed onion skin cells appearing shrunken with a color change commonly seen in a hypertonic solution. On the 25th slide, we are talking about the bacteriology or the study of the bacteria. The first one we have is the Diplococcus pulmonale. Is the one that causes pneumonia in humans. It has a spherical bacterial cells in pairs. That is why it is called Diplococci. Then the second one is the Vibrio coma, a marine bacterium associated with aquatic environments. It has a comma-shaped curved bacterial cells, often with a single flagellum and appearing slightly curved or spiral. The color can vary, but it is commonly gram-negative. Then the third one is the Bacillus subtilis. It is commonly found in soil and associated with a wide range of ecological roles. And the shape is rod-shaped bacterial cells, that is why it is called bacilli, that may form chains with a color that depends on the gram staining, but is often appearing pink or purple. The fourth one, we have the Serratia marcescens, is often found in soil, water, plant-associated environments known for its red pigmentation. It has a rod-shaped bacteria with a pinkish red color, and it is noticeable especially when it is observed under a agar plates. Then the fifth one or the last one is the Streptococcus pyogenes. It causes various infections in humans, including strep throat and skin infections. It has a spherical bacterial cells in chains. That's why it is called the Streptococci. Then moving on the 31st slide, we have some guide questions. Then the first question is that on the slide number 32, which part of the microscope are used to magnify the objects or the specimens? The answer is the objective lenses is the part of the microscope responsible for magnifying the objects or specimen. It provides different levels of magnification at 4 times, 10 times, 40 times, and finally 100 times. So basically each objective lenses that is attached on the nose piece has its own level of magnification. So the low power is color coded with red, which is the 4 times, And then the high power is coded with oil, which is 100 times. So this level of magnification reveals structures that cannot be seen by the naked eye. So the second question is that why is the resolution of microscope is more important than magnification? We must take into account that when we observing specimens which require higher resolution, such as distinguishing the subcellular parts of a microbe, magnification won't stand alone as it will require a higher resolution for us to observe it in a fine detail and for it to be structurally accurate. So by the definition of magnification, we're just zooming in the objects that we would like to see because it cannot be seen by the naked eye. But if the magnification is higher but the resolution is lower, then it will produce a blurred image. So magnification won't stand alone itself. For the third and last question, describe the mirror or illumination part of your microscope. Why would a concave mirror be better than just a plain flat mirror in an illuminating specimen? The answer is that traditional light microscopes, particularly ones without inbuilt lighting, utilize a mirror to reflect light from an external lamp or from the surrounding environment up through the stage's bottom where the specimen is positioned. When it comes to microscope illumination, concave mirrors are preferred over flat ones because of their capacity in focusing light onto the specimen, producing illumination that is brighter, more intense, and as well as more consistent.